Let's begin with the lesson cell, the unit of life. We are all aware of the definition of a cell, that a cell is the basic or fundamental structural and functional unit of all living organisms, which means every living organism's structure basically comprises of cells, and whatever function we perform, actually it's an outcome of the activity of the cell only. A cell could be seen under a microscope. So we can say invention of the microscope only led to the discovery of the cell. The first kind of microscope or the simple microscope was constructed by Antony von Leeuwenhoek, which had only one magnifying lens, whereas after that Robert Hooke constructed a compound microscope having two lenses. The lens which was placed close to the object or which was close to the object is known as the objective lens, and the one away from the object but close to our eye is called eyepiece. So this doubled up the magnification power, and this microscope is known as the compound microscope. Robert Hooke observed a thin slice of cork under his compound microscope, and he found a honeycomb like structure here, just like a honeycomb, which consisted of empty boxes. These boxes were empty because these cells were dead, and all we could see was, or all he could see was, the dead cell walls, but no living protoplasm was inside. So first a simple microscope was constructed by Antony von Leeuwenhoek, and then a compound microscope was constructed by Robert Hooke, and Robert Hooke only discovered the cell under his compound microscope by observing a thin slice of cork. Now only the cell outline and a bit of the cytoplasm and nucleus could be seen under the compound microscope, not by Robert Hooke because he had observed dead cells, but for further magnification an electron microscope was invented whose magnification was 200,000 times much more than that of a compound microscope. So the cell details or the organelles inside a cell could be studied only after the invention or construction of an electron microscope, which uses a beam of electrons which are bent by magnets. Coming up next to the cell theory. We have studied about cell theory before also. The three main scientists who proposed the cell theory were Schleiden, Matthias Schleiden, who was a botanist, Theodor Schwann, who was a zoologist, and Rudolf Virchow, who made an addition to the cell theory. The cell theory was originally proposed by Schleiden and Schwann, and later on Rudolf Virchow in 1858 made an addition to it. The main points of the cell theory are that the cell is the smallest unit of structure of all living things, it is the smallest unit of function of all living things, and all cells arise from pre-existing cells. This third point was added by Rudolf Virchow to the original cell theory proposed by Schleiden and Schwann. Now we have already discussed what it means by calling cell a structural unit and calling it a functional unit. Next, coming to uni and multicellular organisms. Those organisms which are made up of a single cell are called unicellular, like bacteria and yeast, and the ones which are made up of several cells are called multicellular, like human beings or mango or neem plant. A unicellular organism, a single cell does all the work, whereas in a multicellular organism there is division of labor. The work is divided to different groups of cells. Next we talk of the importance of cell being small, how smallness of size of the cell makes it more efficient. For this I made a simple sketch. Like you can see, this is one large cell versus several small cells. Now you yourself can make out that the surface area of this single cell is much less as compared to the surface area of these several small cells which may be occupying the same space or volume. These are several cells and they are all providing a larger surface area as compared to one big cell. So we say that several small cells are much more efficient than one large cell. Why? Because more surface area, greater would be the efficiency. Because in the same volume or in the same space, if surface area provided is more, these cells would be more efficient. The nucleus would exercise a better control on the cytoplasm, and cells would be able to perform their functions more quickly. They would be able to exchange materials quickly, able to take up gases quickly, remove waste quickly, take up nutrients more quickly. The nucleus has a better control over the cell. Even repair of such cells is much more easier than one large cell. Say our body had few large cells and one or two of them get damaged, our working would get impaired. But several cells, it ensures that they are quick in their repair, and even if they don't get repaired, other new cells take their place. So this means that several small cells, or the smallness of the size of the cell, makes it more efficient. That is why our body is made up of several cells. Here the explanation has been given that how the larger surface area to volume ratio would help in better diffusion of nutrients, better removal of waste, better inward and outward diffusion of respiratory gases, and damage to the cell also can be easily repaired. Now cells vary in their shapes, and the shape of the cell is related to the function it performs. Like red blood cells, they are biconcave in shape like this and very small in size. This helps them, the biconcave nature helps them to carry more oxygen, and small size helps them to move through the narrow capillaries. The white blood cells are amoeboid or irregular in shape, and they can keep changing their shape easily. This allows them to throw their pseudopodia out and squeeze out through the walls of the capillaries, reach where the germ is trying to enter our body, and engulf it by throwing their pseudopodia around it. So we are seeing how the shape is it depends on the type of function the cell has to perform. Next is the nerve cells. The nerve cells are long and they have thread-like projections running like wires all over our body, as they have to conduct or carry messages from different parts of the body to the brain and vice versa. So they are long and are thread-like to conduct messages. The muscle cells are long and contractile to pull or squeeze the parts. Likewise, the guard cells, mainly in dicots, they are bean-shaped to open and close the stomata. We have studied about guard cells also. These are the two bean-shaped guard cells which are guarding this opening of the stomata called stoma. So because of this shape only, they can regulate the opening and closing of stomata. So we can easily say that the shape of the cell depends upon the function the cell has to perform. Moving on, next is the structure of the cell. There are some parts which are present in every cell. Three main parts are the cell membrane or the plasma membrane, the nucleus, and the cytoplasm. And the cytoplasm contains large number of organelles. Organelles are tiny living parts of the cell concerned with performing specific functions. Now, if we take a look at an animal and a plant cell, we will find some basic differences. An animal cell has only one limiting layer or surrounding layer called the cell membrane, whereas a plant cell has a cell wall and a cell membrane also. So cell wall is an additional layer present in the plant cell, absent in the animal cell. Moreover, plant cells also contain chloroplasts, which are absent in the animal cells. So two things present in a plant cell and absent in an animal cell are chloroplasts and cell wall. Whereas animal cell contains a part called centrosome, which helps in cell division, which is absent in a plant cell. So these are basic differences between plant cell and animal cell. Some parts are common, like both have mitochondria, both have Golgi bodies, both have endoplasmic reticulum, both have vacuoles, but you will find vacuoles are very small and often temporary in an animal cell, whereas they are large and permanent structures in a plant cell. So if we compare plant and animal cell, These are the differences which we will find. All living parts of the cell, like the centrosome or the Golgi bodies or the mitochondria, this diagram is more detailed. You can practice this one. Here these are the mitochondria, these are the endoplasmic reticulum present in the cytoplasm, or we have lysosomes. They are all tiny living parts of the cell and they are called organelles. There are some non-living parts of the cell also which are called cell inclusions, like the vacuoles or the granules which are present here, the vacuole or any tiny granules present here. They are called cell inclusions. So living parts of the cell are called organelles, and non-living parts of the cell which are temporary in nature, formed as a result of metabolic activities, are called cell inclusions.